Ayun, thank you. Thank you, nag-notify na siya na this is now recorded. Okay, so but anyway, if you have questions, so you can raise it over our chat box. Meron ba? Ayun. Oh, di nga nagano ngayon ni Alexter. Sorry sir for the okay. Sige, take note. Okay, so again, this is pre-recorded, recorded. Ayan. So I hope you can see my screen now. Paano na-share screen tayo? Yan. So, molecular radiobiology. True na ora no di rin yung ma-perfect ini pag chapter test next week. So molecular radiobiology kay again, our human body is composed of 5 major molecules, 4 of which are macromolecules. So we have again the macromolecules. So we have the proteins, we have the, of course, nucleic acids, we have lipids, and we have carbohydrates, and the fifth one, which is water. So these are the 5 major molecules of the human body. Macromolecule in a sense that their structure is quite complex, and water kasi is simple lang, H2O. So again, the effect of irradiation of macromolecule is quite different from that of water. So when, as I've said, ora-ora di rin natin ma-perfect, ano? Pag sumugad kita nga water, iti interaction is called radiolysis. So when macromolecules are irradiated in vitro, so take note, we have the letter T. Diba, sabi natin T, test tube. So that is outside the body or outside the cell. So a considerable radiation dose is required to produce a considerable effect. So sabi natin last time, sa radiation dose response relationship, di ba? Para makaimod kita na talagang magkakaroon or there will be a appreciable effect doon sa structure or sa tissue, kailangan, di ba, linear threshold. So meaning there should be a dose or a radiation dose is required to produce a considerable effect. So there should be a threshold dose. No? So irradiation in vivo, yan. So this is the opposite of in vitro. So meaning that is within the living cell. So demonstrates that macromolecules are considerably more radiosensitive in their natural state. So what is trying to say in this slide is that mas ano daw mas nagre-respond sa effect ng radiation if that is again in vivo, no, that is in their natural state. Pag outside the human body, kailangan mataas yung amount ng radiation para masabi nating may effect on radiation. Kaya somehow if you are conducting a research on this one, so sasabihin mong radioactive, I mean, ano siya, radioactive, radiosensitive versus radioresistant. Kasi this is the concept. Mas magre-respond talaga ang cell, okay, or tissue if this is inside the human body and kailangan natin ng threshold dose pag outside the human body. Okay, I hope you can take note of that. Okay, so irradiation of macromolecules. So a solution, this is a liquid that contains dissolved substances. So parang naging ano tayo dito, na naging chemist. So di ba sa chemistry meron tayong iba-iba yung definition natin, substance, suspension, solution. So yan. So we have a term here, solution. This is a liquid that contains dissolved substances. So a mixture of fluids such as water and alcohol is sometimes or is called a solution. Okay, when macromolecules are irradiated in solution in vitro, again in vitro, that is in a test tube or outside the human body. So these are the observed effects. Ano? Again, these 3 are only observed in vitro. I would like to clarify these are observed in vitro. So first we have the main chain scission. Next we have the cross-linking. and we have the point lesions. So as illustrated here, I hope you can see it in your screen. So this is, for example, let's take this a long chain macromolecule. Let's consider this one as protein. So alam naman natin that there are 22 amino acids. Tama ba yun? There are 22 amino acids in protein synthesis. So, halimbawang may X-ray, okay, ibahin natin yung kulay. So, halimbawang may X-ray, gawin natin color blue. So, ito, radiation. So, mag-interact siya doon sa isang long chain macromolecule. So, what happens is, again, as observed in vitro solution, so pwedeng magkaroon ng main chain scission. Pag sinabing, they're separate naman, I mean, we'll define that later. Okay, so, another thing is pwedeng magkaroon ng so cross-linking. So for example, sa main chain scission kasi, di ba, scission or scissor. So parang ginugupit natin, pinuputol natin ang isang long strand. So yung isang strand na to, pwede siyang dumikit doon sa isang long chain molecules. Yan, parang ano ba? Parang kumakabit. Yan. Ayan, ganun, mas maintindihan kasi niyo pag may mga halong chika-chika eh. Okay, so pag halimbawa, isang chain talaga. Okay, so let's say isang amino acid na natanggal doon sa long chain. So makikikabit siya sa isang another chain, no? So pwede nating masabing this is a cross-linking. So point lesion, for example, may na-observe dito, ayun. Letter C. May mga na-observe dito para mga namamaga, yan, mga nahubag-hubag. So that's a point lesion. So again, these three are the observed, effect of irradiation, again, in vitro. Mahirap makapag-conclude na this is happening inside the human body because the observation is based on in vitro solution. Okay, so let's define all of these things. This main chain scission, this cross-linking, and this point lesions. Okay, so sinabi natin kanina, like for example, in this illustration, sa letter A, okay, so this is a main chain scission. So main chain scission is breakage. Yan, so we have the blue breakage. Okay, so akala nyo kasi may forever. Kaya take note ha, Mr. Bisan naman la online, Justin lagi hapon na napansin. So breakage of a backbone of a long chain macromolecule. So in which sabi in by definition, so masasabi natin yung radiation pala, yung X-ray, kaya niyang buwagin ang matibay na pagsasamahan. Ayun, walang forever. So the result is the reduction of a long single molecule into many smaller molecules, each of which may still be a macromolecule. So, gaya yung sinabi ko kanina, let's take for example, this is protein. So alam naman natin sa protein there are 22 amino acids, no? So if there is the reaction of radiation, so si X-ray pwede niyang buwagin yung long chain macromolecule. Yung 22 amino acids, pwede niyang paghiwa-hiwalayin. Pwedeng isa-isa, pwedeng by group, parang parang the boat is sinking. Group yourself into 4. Yan, pag yung isa walang pair, yan. So, yun. May kita natin, as observed here, this is a long chain macromolecule. After irradiation, yun, left. May radiation siyang binanggit dito, beta. Ayun, we have the right scission. So this is main chain scission. Parang scissor. Yan. Para kang sa mga mahilig mag-art diyan, di ba? Ang mga ginugupit-gupit mo yung gunting. So parang ganun yung reaction. Let's take note that yung papel is the macromolecule, yung gunting is the radiation. So parang hinihiwalay-hiwalay mo yung isang band ng macromolecule. Okay, main chain scission reduces not only the size of the macromolecule, but also the viscosity of the solution. So, for example, viscosity of the solution. Let's say, for example, sipon. So, pag pisnganin mo, malapot, di ba? Parang kinukurean kapag pisnga. So, ansipon, parang taho. Butang matubig, yan. Parang nagiging ano na siya. Parang hindi na siya ganun kalapot. Like for example, in this illustration, sa picture natin. So, masasabi natin, this one, ayun, si picture 1, hindi siya ganun kamalapot as compared with picture 2. Pero if we are going to compare all of these solutions, masasabi natin pinakamalapot yung last na picture kasi hindi nga nag-flow, viscous yung tawag dyan, viscosity. So the more malapot siya, dumidikit talaga yung solution. So the more viscous it is. So, sabi dito, yung main chain scission reduces not only the size, but of course, the characteristic. Halimbawa, yung isang molecule, malapot siya. When it is irradiated, nagiging na lang siya, easy to flow. So, a viscous solution is one that is very thick, slow to flow, such as a cold maple syrup. So, tap water, on the other hand, has a low viscosity. So, measurement of viscosity determine the degree of main chain scission. So, for example, for this one, so pwede nating sabihin if this is the actual molecule that is irradiated, irradiated in vitro, tapos yung effect niya is ganito lang, so sabi natin, hindi ganun kalala yung main chain scission. But if this is the effect, sabi natin, ganun siya naging kamalala yung main chain scission sa isang solution, sa isang macromolecule. Okay, nagets ba guys? So, what happens next is that pwedeng mangyari yung letter B, no? Kasi nasira natin yung matibay na pagsasamahan ni letter A. So, pwedeng halimbawa, di ba sabi natin, ginupit-gupit mo yung papel. So, yung isang papel, mo siya, ipopost mo sa isang papel na naman. So, what happens is that this can become a cross-linking. Ayun. So, parang parang ikaw, may jowa ka na, nangabit ka pa sa iba, na-link ka sa iba, yan. Cross-linking ang tawag dyan. No? Some macromolecules have small, spur-like side structures that extend off the main chain. Others produce this spur as consequence of irradiation. Yan. So, ito, for example, this is the cross-linker. This is a chain of macromolecule. Okay? So, kung mapapansin natin, itong cross-linker, dumikit siya dyan, or ito, dumidikit siya kahit saan. So, this is quite an effect. So, meaning, after main chain scission, yung nakat na structure, pwedeng dumikit sa ibang part. So, these side structures can behave as though they had a sticky substance on the end, and they attach to a neighboring molecule or to another segment of the same molecule. This process is called cross-linking. Yan. So, para mas madaling tandaan, madali man lang remember, I guess, I hope so. Parang labi na't mga girls didang nakaka-relate. Name drop ko na Justin Tejero, kay damot kaaway, charit. Ayan, so, pwede masabi, nagiging, nagko-cross-link siya, nagko-cross yan. So, radiation-induced molecular cross-linking increases the viscosity. Kung kanina, an aton main chain scission, ginbabawasan niya ang pagkamalapot ng substance. Ito naman kasi, di ba, dudugangan mo another molecule. So meaning, mas magi-increase. Kaya, sabi dito, increases the viscosity of the molecular solution. Mas magiging malapot. Kaya madadagdagan na naman ng isang molecule. And of course, si letter C. The letter C, this is point lesion. So, radiation interaction with macromolecule can result in disruption of a single chemical bonds producing point lesions. So, point lesions are not detectable, but they can cause minor modification of molecule, which in turn can cause to malfunction within the cell. Okay, again, sabi dito, point lesions are not detectable. So, hindi natin madaling masasabing, may point lesion siya. Kasi parang parang may adahin, sabi nga dito, modification. Let's say, parang nagkakahubag-hubag. So, palitan natin ng color red. Yan. Let's say this molecule, okay, didikit siya dyan. So, magkakaroon ng, parang yung explanation nito is parang parang nagkakaroon ng, diba? Before maging cancer kasi hubag muna hubag. So, tumor. And then tumor progresses into what we call malignant tumor. So naging cancer. So this one can explain why irradiation can cause cancer. Kasi doon magsisimula ang point lesion. Take note, lesion or hubag muna. Diba? Hubag. So lesion, pwede naman makategory nga tumor, and then when it progresses or metastasize, pwede natin sabihin, ano siya, this is a malignant tumor. So again, from irradiation, from a simple irradiation, let's say, makukonect natin. Kaya masasabi natin, pwede palang magkaroon ng cancer. Yes, because of the point lesion. But take note, guys, these observations are only in vitro solution. Hindi pa natin mako-confirm in vivo solution. So, meaning at low radiation doses, point lesions are considered to be the cellular radiation damage that results in the stochastic radiation effect observed at the whole body level. Low radiation dose, pwede nating masabing, okay, cellular radiation, ano lang siya, talagang nag-respond lang yung, yung cell, di ba? Take note of the 4 R's. If you can still remember the 4R, we have the radiation, the response, the repopulation, and the recovery. So, point lesions are considered to be the cellular radiation damage that results in the stochastic. Sinabi natin stochastic, di ba, late effect, radiation, di ba, this is late effect of radiation observed at the whole body level. So, laboratory experiments have shown that all these radiation effects, okay, all on macromolecules are reversible. So, pwedeng bumalik sa dati yung molecule through intracellular repair and recovery. Again, so hindi forever yung effect na yon. Yung point main chain scission, yung cross-linking at point lesion. So, sabi dito, they are reversible. Pwedeng bumalik sa original na structure ang molecule. Again, through intracellular repair and recovery. Diba? So again, remember the 4 R's of diba? 4R of radiation or radiobiology. Radiation, response, ano pa yun? Repopulation, and then recovery. So eventually, what we are trying to say here is, because the human body is so complex, so unique structure, that we can, okay, that it can recover from amount, okay, from radiation response. Parang may nag, is there anybody who akala ko may nag-notify kasi. Anyway, if you have question, guys, don't hesitate. You can directly interrupt me. Sir, excuse me kasi naka ako yes, Miss Clarissa. Ay, sorry. Kala ko you're raising your hand. So if you have questions, again, do not hesitate. You can to make this online discussion more interactive. I appreciate that. If you have question, if you want to clarify something, so you may interrupt me by saying, sir, excuse me, kasi hindi ko makikita masyado dito kasi naka-share ako ng entire screen. Okay, so again, do not hesitate. Medyo maisuglan ako ng pagka-approach siya na. Pero, ba naman, late ba naman ka? Malilipay pa ako. Charay. Okay, sige. So, let's continue. So, we have the macromolecular synthesis. So, di ba, alam naman natin noon, wow. Alam naman natin noon that when we say synthesis, so we need to, di ba, usang isang cell, before it can undergo mitosis or meiosis, kailangan ma-synthesize muna natin. Diba? Baga, pinapahinog muna natin ang isang cell bago siya mag-undergo ng repopulation or proliferation. Kailangan, pag matawag na natin, mature na ang isang cell kasi nag-undergo na siya ng synthesis. So, macromolecular synthesis. Paano magmamature ang usang cell or an usang molecule. So here, we need to be specific molecule. Paano nagmamature ang isang molecule? Like for example, protein, paano siya magiging matured ng protein? Nucleic acid, paano siya magiging mature ng nucleic acid? Okay, so modern molecular biology has developed a generalized scheme for the function of normal human cell. So molecular nutrients are brought to the cell and are diffused through the cell membrane where they are broken down, which is catabolism, or into smaller molecules with an accompanying release of energy. So this energy is used in several ways, but one of the more important ways is that they are used in the construction or synthesis of macromolecules from smaller molecule, or which is what we call anabolism. Again, so here masasabi nating pinapamature mo na ang isang cell, no, or ang isang molecule. So they are brought to the cell and diffused through the cell membrane where they are broken down into smaller molecular or molecules with an accompanying release of energy. Okay, that's why uh, for molecular or cellular digestion, 'di ba? Kailangan natin, sabi nga natin noon, like for example, if this is a carbohydrates, kailangan natin i-use up ang ating energy. Kaya although I sorry na kasi naka-online lang tayo. Kailangan ano, 'di ba? Kailangan gumagalaw-galaw tayo. Kailangan yung energy natin ma-consume sa body. Kasi otherwise tataba tayo, no? So where they are broken into catabolism into smaller molecules with an accompanying release of energy. So this energy is used in several ways, but one of the more important ways is that they are used in the construction or synthesis of macromolecules from smaller molecules or anabolism. The synthesis of protein and nucleic acids is critical to the survival of the cell to its reproduction. So what we are trying to say here is that before yung cell mag-mature, before yung cell mag-undergo ng division or molecule mag-undergo ng replication, kailangan i-mature muna natin. So let's try to watch this video kung functional pa to. So I'm going to copy this one. Parang may isang pumasok. Or may nawala or ba yung bumalik. Yeah. So stop po na yung presentation ko kadali ha. So let's try if this video is still functional or not, or para at least we can have an idea on what this one, how it looks like.
Feature makes project management a breeze, so you can focus on what really matters, collaborating with your team to achieve your goals. Say goodbye to multiple apps and hello to seamless team collaboration. Click up.
Yeah.
Hello friends, today we will discuss the concept of metabolism. So whatever we eat, whether it contains proteins, carbohydrates or lipids, all these are digested and converted into their simpler form in our body. Proteins are converted into the amino acids. Complex carbohydrates like starch, they are converted into simpler sugars, and lipids are converted into fatty acids. Now during this process, the energy is released in the form of ATP. And other energy rich molecules like NADH2 or FADH2, they are also formed and which can be used for different purposes. Some waste material like nitrogen compounds, CO2, phosphates and sulfates, they are also produced during this process. Such waste material is excreted outside the body by different ways. Now these simple molecules can be again used to produce different complex and large molecules which can play different role in our body. For example, enzymes, the hormones, the receptors and even the hair and nails. So as you can see, whatever we eat, it changes from one form to the other. And in Greek language, the word change, is called as metabolismos. And from this Greek word, the new term came into the existence in science, the metabolism. So, metabolism can be defined as sum of the total of all these biochemical reactions occurring in a cell. And this interconnecting network of reactions are called as metabolic pathways. Metabolism can be classified into two categories: catabolism or catabolic reactions, and anabolism or anabolic reactions. Catabolic reactions are those in which larger and complex molecules are degraded into smaller and simple molecules. And during these reactions, energy is released in the form of ATP. For example, conversion of proteins into amino acids and conversion of lipid into fatty acids. A classic example of catabolism is the degradation of glucose molecule into pyruvate. Glucose is hexose sugar, that is, it is made up of 6 carbon, and pyruvate is made up of 3 carbon, that is triose. This degradation occur in 10 stepwise reactions, which is known as glycolysis. And during this conversion, 2 ATP molecules are produced. Anabolism is exact reverse of catabolism. In anabolism or anabolic reactions, small and simpler molecules, they are converted into large and complex molecule. And during these kind of reactions, energy is consumed. In catabolism, energy is released, and in anabolism, energy is consumed. For example, conversion of amino acid to keratin protein, which is present in the hair and nail. Another example is synthesis of glycogen or starch from glucose molecules. So this was all about the basic concept of metabolism. Thank you very much for watching this video. And if you like this video, please like and subscribe my channel.
Thank you. Okay, so yan, nakita natin guys, ano? So, balikan ko. Stop sharing. Tapos balikan ko natin PowerPoint. Parang again, pasensya lang kay talaga nag-allergy talaga ako at some point. Kaya ayokong pumasok kay damot nagkakatul-katul parang sumugay yun, sir kinakag ikaw pa naman Justin malibak pa naman niya. Dito, so medyo makatul-katul talaga nag-medicine pa ako. So nakita natin naton video ano, so sabi doon, di ba, ang ganda ng pagkaka-explain niya, catabolism from large molecule into a smaller molecule and anabolism from simple to large molecule. So, an energy nga gintawag niya di ba? So, ang iya ginhatag niya energy is the ATP or the adenosine triphosphate. Yan. Gin example. So, Trontano, modern molecular biology has developed a generalized scheme for the function of normal cell. So, molecular nutrients are brought to the cell and are diffused through the cell through membrane where they are broken down, diba? And large molecule into simple, okay, so into smaller molecules, like for example, and we know for a fact that, like for example sa video, we know for a fact that the protein, protein has 22 amino acids. So ano sang molecule ng protein, pwede niya i-broken down into 22 amino acids. So that is catabolism. So an energy i-release in a form of ATP. And alam naman natin guys, di ba, kung hain kin which part of the cell kin synthesize ang protein, di ba, doon sa ribosome. Yan. So again, with this, the accompanying release of energy in a form of adenosine triphosphate. Paki-remember ya, bayan makasuod ka mo na liwat nga. Wala man inin among discussion and kaya nung made a chapter test or a discussion. So wala aton bukan no, pero that's the basic concept of protein synthesis. So this energy is used in several ways, but more of which important ways is they are used in construction or synthesis of macromolecule from smaller, or that is anabolism. So the anabolism is the opposite, di ba? So the synthesis of protein and nucleic acids is critical to the survival to the cell and to its reproduction. So pag sumuad kita liwat ng anabolism from small to large, so meaning an ATP gagamiton naton, na-release kanina ng energy gagamiton naton for production of large molecule. Okay, so proteins are manufactured by translation. So later we'll watch this video again. So let's talk about proteins muna. Alam naman natin that one of the major molecule of the human body is the protein. Diba? Protein is composed of 22 amino acids. Okay, so they are manufactured by translation. So we will discuss paano ginihimo ang protein or how they are synthesized. Diba? Saan? Sa specific part ng cell, doon sa pangit ng pagkasulat ko kasi wala, ribosome. Yan. Doon yung site of protein synthesis. So unang segment ng protein manufacturing or protein synthesis is translation. So of the genetic code from RNA, the ribonucleic acid, or this, of course, kaya maraman kita nga, di ba si DNA, okay, si DNA ito pinaka mastermind. Okay, so genetic code transfer or transfer RNA, which had been transferred from the messenger RNA. So the information carried by the mRNA was in turn transcribed from the DNA. So si DNA talaga pinaka-mastermind, siya pinaka-controlling part ng cell eh. So tanan niya message, so papaagi niya ka mga RNA. So una again, translation. Kung anong message niya, so ipapatranslate naton, ipapatranscribe naton gamit din ng mga RNA. Next. Okay. Sige, ano eh. Higdanta muna di ka video. Kaya ba ngayon? How this sorry, wala na. Hindi na functional yung video na yan. Parang ginatanggal na ni YouTube. But anyway, balik ako ulit. So again, para makahimo kitahin protein, so kailangan may da muna message tikang kan DNA. So ano message, ita-transcribe para makikichika, no? So kailangan may mga messenger muna ng transfer RNA down to the ribosomes. Or next, we have from the molecular synthesis. So radiation damage to any of these macromolecules may result in cell death or late stochastic effects. Proteins are continually synthesized throughout the cell cycle and occur in much more abundance in nucleic acids. Okay? So they are interrelated, no? So from the amino acids, so kailangan makaproduce kita ng mga proteins, okay? Para manatong and major composition, the DNA is inside the nucleus. Okay, so furthermore, multiple copies of specific protein molecules are always present in the cell. So consequently, proteins are less radiosensitive than nucleic acids. So it says here that proteins play essential role in the cells of all living creatures. They serve as building blocks of cells, control chemical reactions, and transport material from the cells. So proteins are composed of long chains of amino acids. So the specific sequence of amino acids in a chain determines the exact function of the protein. Yes, because as a review, we can, di ba, na-mention natin that protein acts as enzymes, acts as hormones, and okay, so they act as, okay, the primary defense of the body. Okay. So similarly, multiple copies of both types of RNA molecules are present in the cell, although they are less abundant than protein molecules. So on the other hand, DNA molecule with its unique assembly of bases is not so abundant. That's why we can say that DNA is the most radiosensitive molecule. Kaya no, pag mamatayan, pag ma-irradiate ang DNA, patay ang bugos na cell. No, so that's the key. That's a cue. So the deoxyribonucleic acid is a very long chain molecule that is densely packed. So ordinarily, its molecular structure would be difficult to analyze using crystallography. However, it can be prepared for X-ray crystallography by extracting it from cells in form or gel form, and then slowly drawing into fibers in which the molecules run parallel. So we know naman ano how the DNA looks like. So it looks like a we call that one the double helix structure. Check ko lang kung naka-share na akong screen. Okay, so DNA is synthesized somewhat differently from proteins. So during G1 portion of the interphase, deoxyribose, phosphate, and base molecules accumulate in the nucleus. So these combine to form a single large molecule that during the S portion of the interface, this is attached to an existing single chain of DNA. And during the G1 molecular, DNA is in the same familiar double helix form. Okay, so let's watch this video muna. So hopefully it is functional. Hindi pa nagtanggal doon sa YouTube. Hanap na lang tayo ng, ng mga videos dito. That's quite related. Protein synthesis. It's ayun. Okay, so let's watch a video muna for protein synthesis and for the DNA sequence. Yun na, the one.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, If you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So, the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, thank you so much, Amiga Sisters. Yan. So I hope you can relate to our discussion. Talagang it's quite complex to put all the concepts here. Kay iba din supposedly when you were in high school, wow. Ano yun? So at least dito na nalaman natin, di ba yung, sorry, I need to share my screen. Again, challenge, stop share it. Ayun. So thank you so much. This is the perks of online classes kasi I can share all of my some of the instructional materials from YouTube into one. And later on I can send the recording of this lecture para magamit manliwat niyo para at least ma-review niyo kahit paano. Okay. Share my entire screen. Okay, so at least here we at least with the help of the Amoeba Sisters na-define natin so the two steps, the translation, the transcription first and the translation. Of course, it cannot we said that the DNA is the mastermind of siyang nagko-code, but hindi ma-accomplish kung walang mga messenger at saka walang transfer RNA. Okay, so what else? Next, we have the DNA synthesis. It's quite different. As I said, it's quite different from the protein synthesis. Sige na, let's watch this video. I hope you'll not get bored on this one. Di pa naman ano. So try natin. Charan, stop share. Yeah. So let's watch this video again from the Amoeba Sisters. It's quite helpful din naman for the Hindi ka, ano. Ayun.
Tatanggalin ang bad odor.
This course is about systems and system thinking.
DNA. We talk about it so much, it's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where. Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when. When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, The sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors. In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions and the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So ayun, di ba? Ang ganda ng mga pinagsasabi ng mga Amoeba Sisters. Tapos parang feeling ko madadamo na lalo yung studyhan naman. I'll help you arat man para next week. At next week, may klase ba kita next week? I don't know. Wala pa man ganun nga, wala pa man ako ganun nga memo or announcement. But anyway, mag-prepare na lang kita no kay it's quite interesting. It's getting interesting because this is molecular radiobiology. Damong mga pinagpapasok ng mga concept, di ba? Like we have the codon, di ba? The different function ng mga enzymes, di ba? We have the helicase, the polymerase, the primase, and the ligase. Yung mga tinatawag na leading strand, lagging strand, yung Okazaki fragment, yung mga semi-conservative. So I hope you can get to have a recap on that thing because as I've said, these are the background for molecular radiobiology. So if there are classes next week, again, we can have a chapter test on this one. Of course, this is on the molecular radiobiology. Okay, so let's continue. So I hope you can get a gist on this, guys. Medyo nakuha natin yung concept. Bakit nagkakaroon ay how the protein are synthesized, how the DNA are synthesized. So that's the key why we are getting some information, some input, video, helpful instructional materials from YouTube. Buti na lang may YouTube. So thank you, YouTube, and thank you, Amoeba Sisters, for the wonderful insights. Again, don't worry, I'm going to send the recording later for you to be able to have a recap on our discussion. So, paki-check na lang sa ating Google Classroom. Okay, so that's how the DNA synthesis based on the video. Again, DNA is synthesized somewhat differently from protein. Although both of them are essential. So take note may dadalawang cell although hindi nasabi sa busyong. So may dadalawang cell, of course, the eukaryotic and prokaryotic. Yung isang cell which has a nucleus and isang cell na walang nucleus. The eukaryotic may mga nucleus, the prokaryotic wala. Okay, so during the G1 portion of the interface, the deoxyribose, the phosphate, and the base molecules accumulate in the nucleus. So these molecules combine to form a single large molecule that during the S portion of the interface, this is attached to an existing single chain of DNA. And during the G1, molecular DNA is then in a familiar double helix form. So half as much as DNA is present in G1 as in G2. So kanina medyo na-explain naman yan, di ba? So as the cell moves into the S phase, the ladder begins to open up in the middle of each rung, such as like a zipper. Now the DNA consists of only one single chain and no pairing of bases. So I don't know if this is here. As I've said, I'm going to seek help from this ito ng mga important na mga Okay, so let's try this one. Okay, stop share ko muna.
DNA replication, the process simplified. The DNA helix is unwound. In leading strand synthesis, DNA synthesis progresses in a 5' to 3' direction. Lagging strand synthesis is a bit more complicated because the DNA is exposed in a 5' to 3' direction. A short RNA primer is synthesized. The RNA primer is extended in a 5' to 3' direction with short DNA segments called Okazaki fragments. As the fork extends, the process repeats, forming a continuous leading strand and multiple Okazaki fragments. On the lagging strand, the RNA is replaced with DNA. DNA fragments are joined. As the replication fork moves on, leading and lagging strands twist into helical forms.
So ganun lang naman kasimple Okay, so thank you. Okay, so yon. So that is dito kasi sa ating reference sa busyong, hindi kasi masyadong na-detalye, especially yung mga leading strand at mga lagging strand. But again, half as much DNA is present in G1 as in G2. So again, half as much DNA is present in G1 as in G2. So as the cell moves into S phase or the synthesis phase, the ladder begins to open up in the middle of each rung, much like a zipper. So salamat sa helicase, talagang taga-unzip. Now the DNA consists of a single chain and no pairing of bases. Okay, so di ba, kanina na-mention naman that yung from the direction of 3' to 5', and then yung isa which is direction of 5' to 3', tapos magkakaroon ng Okazaki fragment, and then leading on, so each of the leading and lagging strand, they twist to form the helix form, no? So yun. Now let's have the radiation effects on the DNA. So DNA, again, is the most important molecule in the human body because it contains the genetic information for each cell. So siya yung mastermind, sabi nga kanina sa video. Each cell has a nucleus. Again, guys, take note, we are referring only for the eukaryotic cells kasi yung iba hindi naman visible yung nucleus, which is hindi naman ganun ka na-appreciate, especially sa mga prokaryotic cells, wala silang nucleus. So each cell has a nucleus that contains DNA complexed with other molecules in the form of chromosomes. So chromosomes therefore control the growth and development of the cell. These in turn determine the characteristics of the individual. Kasi nga, nandoon ang ating genetic code with this DNA. So if radiation damage to the DNA is severe enough, visible chromosome aberration may be detected. Later we will, I don't know if saklaw dito sa sa ating discussion yung chromosomal aberration. So this may be detected. So the DNA molecule can also be damaged without the production of chromosome aberration. Although such damage is, sabi nga dito, reversible, so it can lead to cell death. Pag mamatay yung cell, hindi na yan may-reverse. So, although, again, yung mga na-mention nating mga damages, pwedeng ma-reverse, such as yung mga main chain scission, yung mga, cross-linking, yung mga point lesions. So, pwede daw ma-abrupt yung chromosome formation. No? So, if cell or enough cells of the same type respond similarly, then a particular tissue or organ can be destroyed. And that describes the cause of a deterministic. Again, when we say deterministic, that is an early effect of radiation. So damage to the DNA can also result in an abnormal metabolic activity, uncontrolled rapid proliferation of the cell. This is the principal characteristic of radiation-induced malignant disease that describes the stochastic effect. Again, when we say stochastic effect, this is the late effects of radiation. So, if the damage to the DNA occurs within a germ cell, pag sinabi nating germ cell or germinal cell, diba, these are for reproductive cell, then it is possible that the response to the radiation exposure will not be observed until the following generation or even later. The next generation daw. So, halimbawa, yung anak mo, doon pa lang ma-observe yung effect ng radiation. So, this describes the cause of a genetic effect of radiation. Next, the chromosome contains miles of DNA. Therefore, when a visible aberration does appear, it signifies a considerable amount of radiation damage. So unobserved damage to the DNA also can produce response at cellular and whole body levels. These types of damage that can occur in DNA are as follows. So ito yung mga iba-ibang effect, not necessary na not necessary that these are in vitro. So we can say that these are now in vivo. So pwede nating matawag that there are main chain scission with only one side rail severed. Take note, guys, that that the DNA is double helix. So sabi dito, only one side rail severed. Main chain scission with both side rails severed. Main chain scission and subsequent cross-linking. Pwedeng isang ma-break tapos papunta sa isang strand. Rung breakage causing separation of bases and change or loss of a base. Although each of these effects results in a structural change in a DNA molecule, they are all reversible. Again, so note of the 4 R's in radiobiology: radiation, response, repopulation, and recovery. That's why this is reversible. And in some of these types of damage, the sequence of bases can be altered. Therefore, that code of codons, nag-apply na dito yung codon. Sa ibang mga discussion natin, hinahanap natin to. Ano yung codon? May not remain intact. So this represents a genetic mutation at any level. Diba yung code? Sabi, yung may mga codon chart pa nga ni, di ba kanina? Nag-mention na itong mga amoeba sisters. Okay, so the fifth type of damage or loss of a base also destroys the triplet code and may not be reversible. So this type of radiation damage is a molecular lesion of the DNA. These molecular lesions are called point mutations, and they can be of minor or major importance to the cell. And one critical consequence of point mutation is a transfer of incorrect genetic code to one of the two daughter cells. That's why, halimbawa, kung tapos na-irrigate or na-expose radiation, so natin matira, pagka may mutation. So, halimbawa, unborrowed, pwede siya mag-anak kan cyclops, kan jean gray, kasi salaan, sayupan aton because na-enter genetic code because of the radiation response, no? So that is the effects of, especially if this is a germ cell or a genetic cell. So the three principal observable effects that may result from irradiation of DNA are, first, cell death. Halimbawa, pag mamatay, din at ma-reverse. Diba? Because of this wrong genetic code, so pwede natin pwede mamatay ang cell. Pwede mag-cause cancer. No? And pwede genetic damage. So, bangin pagkaanak ang daw because of radiation, and then, halimbawa, pwede magkaanak kinalawang ni Cook. Tapos sila direkto sila asawa ng lalaki. Ibang lalaki. Masugaran asawa, di ba ko ibang lalaki? Wala. But it's because of the radiation effect. So the two latter effects, the malignant and the genetic, at the molecular level results in stochastic, so late effect of radiation and conform to, again, linear non-threshold response relationship. So ngayon, we have a radiolysis of water that we are going to discuss. Sige, halipot mo lang ngayon. So, kasi nga, sabi nga natin kanina, medyo iba talaga interaction ng water, or iba ang effect ni water as compared to the other molecules. So, pinasok natin dito yung radiolysis. Pag makabasa nga ni kamoy ni class ng radiolysis, so this is now radiation, or this is the interaction of radiation in water. So take note that, again, human body is constituted, composed of 80%, tama ba yung grammar po? Composed of 80% water. So, because the human body is an aqueous solution that contains approximately 80% water, irradiation of water represents the principal radiation interaction within the body. So, radiolysis, again, is the principal irradiation interaction of the body. Okay, yung kanina, natin yung magnetization, pointation. In vitro man to, diba? In vitro. So, when the water is irradiated, it dissociates into smaller or other molecular products. This action is called radiolysis of water. So, when an atom of water, H2O, is irradiated, it is ionized and dissociate, bubulag, hiwalay into two ions. So this water in the human body is radiation. After radiation, yung water mabe-break down into HOH. So we have the positive ion and we have one negative ion. Okay, so yun ano. So water in the human body, this is radiation. So yung magiging effect nito, yung water, ihihiwalay sa dalawa. Isang positive ion, HOH, and isang negative ion, which is an electron. And after this initial ionization, a number of reactions can happen. First, the ion pair may rejoin into stable water. Yan, take note. Pwedeng yung ion, isang ion pwedeng ano, um, join sa water. Like for example, H2O plus the negative ion, so magiging HOH negative ion. So, ano na lang guys, take note, ganito, di ba sa chemistry, kailangan balance yung kung ilan yung, halimbawa, kung ilan yung hydrogen sa side na to, let's say sa left side, kailangan ganun din ang number ng hydrogen dito sa right side. So, kung dito meron tayong dalawa, so dito, bilangin natin, meron naman tayong dalawa pa din. So, negative lang siya kasi meron tayong na electron. Kanina kasi positive siya. So, bilangin natin, dalawa yung hydrogen. So, sa left side. Dito, dalawa pa din naman yung hydrogen. Kasi lang siya kasi humiwalay ang isang electron. No? So, mas madami yung proton sa side. So, in this case, it occurs. Second, if this ion do not rejoin, so it's possible for negative ion second electron to attach to another molecule. So the following reaction proves yet a third type of ion. So we have tatlong ion na. So 1, 2, tapos we have the third ion, this one, the HOH negative ion. Okay, so the HOH positive ion and the HOH negative ions are relatively unstable and can dissociate into still smaller molecules as follows. So yung HOH positive, pwede siyang mahiwalay. Okay, 1, 2. So H2O, okay? So, and another, the HOH negative, 1, 2. The hydrogen is 1 and 2. So we have here, so may mga asterisk na sign na pwedeng ma-produce. Aside from may mga ions, so we have this asterisk na mga signs. So the final result of radiolysis of water is the formation of an ion, the H positive and OH negative. And yung mga tinatawag natin kanina, yung may mga asterisk, which are free radicals. So the ions can recombine, therefore no biologic damage will occur. So a free radical, this is an uncharged molecule that contains a single unpaired electron in the outer shell. So dito mag-start ang ating mga adverse effect of radiation because of these free radicals. Again, anong mga tinatawag na radicals? Dalawa lang naman, H asterisk and OH asterisk. So the free radicals are another story. They are highly reactive. Free radicals are unstable and therefore exist with a lifetime of less than 1 millisecond. During that time, however, they are capable of diffusion through the cell and interaction at the site. So radicals contain gas energy can be transferred to other molecules to disrupt the bonds and produce point lesions at some distance from the initial ionizing event. So take note, again, ito yung adverse reaction na nangyayari kasi the human body is 80% water. So if these free radicals move from one place to another, so parang ano ba, diri affected or diri exposed an organ sa radiation, but because of these free radicals, they can cause effect or no. Halimbawa, Justin Pero, medyo nag-away. Tapos, mag-benching up man ni Justin Pero ngayon si Rica Reyes. Pinaka-apektuhan si Rica nag-post sa Facebook, ganun. Parang ganun yung so irradiolysis ng water. So, diri Rica, si Reyes and away sa jowa. Pero siya ang pinaka-naapektuhan because Justin, parang siya yung free radical na naka-apekto kan Rika. Nagkaroon ng point lesion doon sa site. So, may mga organ sa ating human body that those are not exposed to radiation. But these organs are ganun, apektuhan na radiation. Kaya because of the free radicals brought about by the radiolysis of water. Yes, some organs are not actually part of the irradiation, but nga kay may mga nagka-cancer man itong organ, it's because of the radiolysis of water. So the H free radical and the OH free molecules are not only free radicals that are produced during radiolysis of water. So here, the OH free radical can join a similar molecule to form hydrogen peroxide. So duha nga OH free radical, so produce tayo. So 1, 2, dalawang oxygen, 2, dalawang hydrogen, H2O2, so hydrogen peroxide is poisonous to the cell and therefore acts as a toxic agent. No? So yun, pwedeng mangyari sa human body. So the free radical, the H free radical can interact with molecular oxygen to form the hydroxyl radical. Okay, HO as, okay, free radical. Okay, so the hydroperoxyl radical along with the hydrogen peroxide is considered to be the damaging product after radiolysis of water. So yun, hydrogen peroxide can also be formed by interaction of two hydroperoxyl radicals. So H2O2 plus O2. So some organic molecules symbolized as RH, dere ni red horse dere red horse, again, organic molecules can become reactive free radicals. So we have organic molecule plus radiation. So it can become a free radical and can be dissociated into two free radicals. So when oxygen is present, yet another species of free radical is possible. Yan. So free radicals are energetic molecules because of their unique structure. This energy can be transferred to DNA, and this can result in bond breakage. So that's why ganun ang effect pala ng radiation sa water. But take note this again, this can only happen pag mga, take note, and mga deterministic and stochastic effect. So ang iba again, deterministic strong taas na radiation dose. Meaning this is a prolonged exposure. Kaya guys, diri maupay nga halimbawa sa mag-X-ray ka, permanente ka lang magpapa-expose to radiation. Ay kay Radtech gusto kong makatanggap ng Bayaning Pilipino Awardee. Wow. So sige-siging ka-expose, take note that our human body is composed of 80% water. Later on, pwede natin makamay adverse reaction effect, pwedeng pwedeng cell death, pwedeng malignant disease, or pwedeng genetic code, or genetic disease. So when biologic material is irradiated in vivo inside the body, a harmful effect of the irradiation occur principally because of the damage to a particularly sensitive molecule such as DNA. So evidence for direct effect of radiation comes from in vitro. Yes, kay malanaton gin testingan direct effect. So wherein various molecules can be irradiated in solution. So the effect is produced by ionization of the target molecule. So if the initial ionizing event occurs on the target molecule, tapos dito mo makita an effect ng radiation, then we can say this is a direct effect of radiation. So halimbawa, ganun na lang, Justin naman, Tejero, parang feeling ko masikat ni talaga ta klase. So, siya ang ginawayan niya jowa, siya talaga pinaka-affected. So, that's the direct effect of radiation. So, an exposed mo is, let's say, an ovary na exposed. An ovary na kami din, an patient na kami din ovarian cancer. So, we can say that is the direct effect of radiation. On the other hand, if the initial ionizing event occurs in this non-critical molecule, which then transfer energy of ionization to the target molecule, indirect effect occurred. So free radicals with their excess energy of reaction are the intermediate molecules. So they migrate to the target molecule and transfer their energy, which results in damage to that target molecule. So the principal effect of radiation is indirect effect. So, halimbawa, kagaya ng sinabi ko kanina, si Justin Tejero, ginaway niya jowa, pero pinaka-apektuhan, Hirika. So, that's an indirect effect of radiation. For example, ang na-X-ray is, ang gin-X-ray mo is, let's say, abdomen, pero nagkamayada siya yung liver cancer, liver, ito man doon. Sige, ibang ta. So, let's say, siya, nagpa-X-ray siya ng chest, pero sige, pag pina-expose, grabe naman, let's say, nagparansera siya sa kanyang, breast. So, nagka-develop siya ng liver cancer, so that can be an indirect effect of radiation. So, it is not possible to identify whether a given interaction with the target molecule resulted from direct or indirect effect. However, because the human body consists of approximately 80% water and less than 1% DNA, it is concluded that essentially all of the effects of irradiation in vivo results from indirect effect of radiation. So when oxygen is present, take note that oxygen is a major cell sensitizer. As in living tissue, the indirect effects are amplified because of the additional types of free radicals that are formed. So that concludes our discussion for today. So ayun. So next week I won't promise that I cannot give, especially kung may klase, I can give a chapter examination based on of course, our discussion, the molecular radiobiology. So do you have any question, guys? Tap share. I hope you get something from this discussion. Hello, ada pa kamo bay nagkaklase na kamo ibang subject. Paki-inform na lang para ako dito sirang plaka. So let me let me check the attendance first, of course, by Marty ako pag nag-attendance, kailangan naka-open cam mo. Tapos maximum naman 20 ko man lang yan. Sige, can we check your attendance first? Kung naada talaga ka mo. Open cam, open fact. Wala nga ni Rabalay, wala nga kamo klase ya pa ipa. Sana all. Parang feeling, wala naman didikuan ni Alexis Tero. Wadi. Ako pa naman taton sikat yan kay indirect effect of radiation. Okay, sige na. Mapaypahikuan ni Christine Shervo, ready na siya para klase niyan. Okay, sige. 1, 2, 3. Kuan tihero, di rin man na-abrit iya camera, pamati ko, at tugulo niya tiya jowa. Ay, ito na. Ada ngayon. Sige. Okay. Smile. 1, 2, 3. And chiring. Okay, sige. So, if you don't have any question, thank you so much. So, just prepare next week. Kung may klase, we have a chapter examination. Kunwari, nalapag-study. Okay? Sige, so, recording stop. Okay, so if you don't have any question, so then I am going to parang pinurian ako, I have to dismiss this class. Okay, thank you so much. And by Friday, ganun pa din, naka-online pa din ako because 5 days isolation. But I'm getting well naman so far. Effective pa naman medication na ako. Okay pa naman mga free radicals, they are still functioning on me. Okay, so thank you so much. And sigurado ka mo question. So kung wala pakiana, then again, we are dismissed. Thank you so much. You can now